Hey guys, today we're going to talk about Chapter 6, the ecological importance of microbes. This might be one of my favorite chapters. All right, so without bacteria, life as we know it would not exist. Bacteria are necessary for nitrogen fixation, for the carbon cycle, and for sulfur and phosphorus cycling. Now, the carbon cycle, I'm sure you guys are all slightly familiar with, right? In order to reproduce, most organisms need to obtain or make nutrients based on carbon, the fundamental element of all organic chemicals, which we are largely composed of and utilize for energy. The start of the carbon cycle is autotrophy. Primary producers, autotrophs, these are organisms that convert carbon dioxide in the atmosphere into organic molecules. Cyanobacteria are prokaryotic autotrophs. Algae and plants are eukaryotic autotrophs. Now, if we continue the carbon cycle beyond autotrophs are the heterotrophs, the consumers. These utilize organic materials and they release carbon dioxide gas as a byproduct of cell respiration. They incorporate organic carbon made by autotrophs into their tissues. Most of the carbon remains in us until we die. And we rely on the activities of these primary producers. Decomposers are heterotrophs that digest and utilize the carbon in the remains of primary producers and consumers when we die. And bacteria and fungi play a major role in decomposition. They also release CO2 gas into the atmosphere as a byproduct. The release of this carbon dioxide gas starts the cycle all over again. And worldwide, the rate of CO2 production far exceeds the rate at which it's being incorporated into organic materials. Why? Well, let's look a little more closely at the carbon cycle. So we've got our atmospheric CO2 and CO2 dissolved in water like oceans and lakes. These things are fixed into organic carbon by autotrophs, plants, algae, photosynthetic bacteria, and protozoans, and chemoautotrophic bacteria. Now, some of these things die and are broken down by decomposers. Some of these things are consumed by other organisms, heterotrophs like animals. Animals release CO2 when they do cell respiration, or when they die, they release methane gas, which goes into the atmosphere, or they're broken down by fungi and bacteria into CO2. But there's another reservoir here, fossil fuels. Now, it used to be 5,000 years ago that once a carbon atom made it into a fossil fuel reservoir, it stayed there indefinitely. Once you became crude oil underground, that's where you were stuck, or coal, or natural gas, whatever. But now humans are mining these things from deep under the earth, and we're burning them through combustion, and we're releasing all that stored carbon as CO2 gas into the atmosphere. We'll also use a lot of these fossil fuels to make plastics and other synthetic products. Unless those things are burned, they don't end up into the atmosphere. But this is a huge source of carbon dioxide gas going into the atmosphere much faster than autotrophs can pull it out of the atmosphere or the water. All right, let's move on to nitrogen. Nitrogen is another important nutritional element required by organisms. Proteins and nucleic acids are all based on nitrogen, and without them, they can't be made. Most nitrogen is found as N2 gas. In fact, it makes up 70% of the air we breathe, but it's not usable by living organisms. Most living organisms, I should say. There are certain types of prokaryotes that are capable of fixing that N2 gas in the atmosphere into forms that can be used by other organisms. Since N2 gas is not a usable form for us, we have to ingest plant and animal material to get our nitrogen. So certain prokaryotic organisms are capable of converting that N2 gas into a usable form. Only prokaryotes can do this, and not all prokaryotes can. During nitrogen fixation, a process in which nitrogen gas, N2, is reduced to form ammonia, NH3. Ammonia can be incorporated into the cellular material of plants. It's mediated by the enzyme nitrogenase, and it requires 16 molecules of ATP for every 1 nitrogen fixed. Energetically, it's very costly to do. Organisms capable of doing this are called diazotrophs. They're free-living, or they could be symbiotic and have an association with plants like legumes. Some examples of legumes are alfalfa, clover, peas, beans, peanuts. These are all plants that are high in protein. Protein requires a lot of nitrogen. Ammonification is when bacteria not capable of nitrogen fixation and fungi decompose waste and dead organisms and use the process of ammonification to obtain nitrogen. Ammonification happens in the decomposition process, and it converts organic nitrogen, or amino groups from proteins, into ammonia, NH3. In most environments, ammonium, NH4+, is formed, which organisms can also absorb. So you see the amine group down here, an amino acid. As those proteins are digested, they form ammonia, which is what makes animal waste smell so strongly. All right, nitrifiers are organisms that can oxidize that ammonium, that NH4+, into nitrite and ultimately into nitrate. Plants use nitrate as a nutrient for growth, which is why it's often listed as one of the important ingredients in fertilizers. Denitrification converts nitrate into nitrite into N2 gas or N2O gas. Some Pseudomonas species are capable of doing this. This is an anaerobic or no oxygen process. All right, and here's a general diagram of the nitrogen cycle. So let's start with plants. Up here we have nitrogen-fixing bacteria living in legume root nodules, like on this bean plant. They're able to fix nitrogen. So nitrogen can be consumed by an herbivore here, the rabbit. It can be decomposed then when the rabbit's done and gone by fungi and bacteria, and those things can be assimilated. So as free nitrates and nitrites, they can be assimilated into new plant matter and move back up the food chain that way. We also have nitrogen-fixing bacteria free in the soil, and they can participate in ammonification. Nitrification, nitrifying bacteria, and some of these nitrates and nitrites will be broken down by denitrifying bacteria to return them up to the atmosphere as N2 gas. So there are some really useful applications of microbiology in the environment. One of these is bioremediation, and it takes advantage of microbial adaptations to use organisms to clean up toxic or hazardous waste by degrading them into harmless compounds. Most known application of this use is for bacteria to clean up oil spills. There are two types of bioremediation: natural bioremediation or biostimulation, where microbes are encouraged to degrade toxic substances in soil or water. We might have to add nutrients to stimulate microbial growth in these areas to get them to do what we want them to. Another type is artificial bioremediation or bioaugmentation. And this is where we actually add the microbes that we want to get to work in an area that are not normally part of that population. Genetically modified microbes can degrade specific pollutants. So some sources of these pollutants might be domestic or industrial waste, dumped into the environments for convenience or accidental spills. Think of dioxin in the river from Dow or Exxon and BP oil spills. Now, synthetic compounds with similar chemical compositions and structure are more likely to be degraded. Xenobiotics are synthetic compounds that are totally different than what occur in nature. These types of things often persist in nature for long periods of time, since organisms are not likely to have enzymes necessary for breaking them down. All right, so here's a comparison. We've got on the x-axis of this graph time in days. On the y-axis is the relative herbicide concentration. So both the yellow herbicide and the purple herbicide start at 100%, right? And we follow them over time. The purple microbe, or the purple pollutant is unchanged over time, right? After 25 days, there's still just as much of it there as there was on day one. But the yellow herbicide is able to be broken down over time so that after 10 days, there's maybe 15 or 10% of what there was at the start. So the purple one would be an example of a xenobiotic. All right. And here you can see oil spill on the left and oil spill on the right that have been treated. On the right side's been treated with bacteria for bioremediation. It is remarkable how well they do. All right, so another form of biodegradation that you can do in your own backyard is composting. Natural decomposition of organic solid material. Mixing garden debris and kitchen organic waste. Exclude meats and fats. But you need to have a carbon to nitrogen ratio of roughly 2 parts carbon to 1 part nitrogen, or 2 part brown stuff to 1 part green stuff is a general rule of thumb. Microbial metabolism causes the pile to heat up to 55 to 66 degrees Celsius, and pathogens are killed at these temperatures. If the pile is frequently aerated, the composting can be completed in 6 weeks. Compost is used as fertilizer and it reduces landfill waste. All right, so that's it for Chapter 6, guys. If this is a topic that really interests you, you might also consider looking into some environmental studies courses.